Biochemical characteristics for identification of anaerobic bacteria. Biochemical characterization is the bacterial identification based on biochemical analysis. It involves determination of biochemical properties of a bacteria. Starting with the Clostridium species, most of them ferment glucose with production of acid. Some also ferment lactose and sucrose. There is no indole production and they can reduce nitrate to nitrite. Some liquefy gelatin. None of them produce oxidase and catalase. Clostridium tetani does not ferment any carbohydrate and proteins. First is the sugar fermentation test. In this test, the ability of bacteria to form organic compound by metabolizing certain carbohydrates and related compounds is tested. ANDRADE'S fermentation broth produces acid as a metabolic byproduct when inoculated with bacteria that is capable of metabolizing the constituent substrate. Two types of fermentation takes place, that is lactic acid, homolactic fermentation, and heterolactic fermentation. Acid production causes a decrease in pH, which results in the color shift in medium. The media appears pale pink prior to inoculation and turns pink to red in presence of acid. ANDRADE'S indicator include acid fusion, sodium hydroxide 1 normal, and demineralized water. ANDRADE'S fermentation broth includes ingredients per liter of deionized water, that is peptone 140, sodium chloride, ANDRADE'S indicator, and distilled water, which is pre-sterilized. Carbohydrate is added by 1% to total volume, which is again pre-sterilized by sterilization. Procedure: inoculate the ANDRADE'S fermentation broth containing carbohydrate of choice with a pure culture of the organism. Add a layer of liquid paraffin over the media to maintain anaerobic condition. Incubate at 37 degrees Celsius and examine daily for up to 7 days. The appearance of a pink to red color in the broth is an indication of acid production. Use an uninoculated tube of ANDRADE'S fermentation broth of the same carbohydrate as a control. This picture is showing the positive and the negative result, where carbohydrate fermentation, thus acid production, is indicated by the presence or appearance of pink to red color in the broth within 7 days following inoculation. A negative result is noted when the medium remains pink, pale pink, or turns yellow. Second biochemical test is NAGLER'S reaction. It is to test the capability of microorganism to make the enzyme lecithinase. The microorganism is inoculated on the medium containing human serum or egg yolk that contains lecithin. The plate is incubated anaerobically at 37 degrees Celsius for 24 hours. Colonies of Clostridium perfringens are surrounded by a zone of turbidity due to lecithinase activity. This effect is specifically inhibited in the medium containing Clostridium perfringens anti-serum containing alpha antitoxin. The positive tests show marked decrease in the opacity of the antitoxin portion of the plate as a result of neutralization of lecithin due to the antitoxin. And negative tests show no loss of opacity zone on the antitoxin part on the plate. Third biochemical analysis is reaction on litmus milk. Litmus milk contains skim milk, that is milk without fat, that contains lactose sugar and casein protein, and litmus indicator, which is an acid base and redox indicator. Reaction on litmus milk. Acidic reaction takes place when lactose as milk sugar gets fermented to acid, and the litmus indicator turns the color to pink. Basic reaction takes place when casein, that is milk protein, gets digested to amines, and the litmus indicator caused the change in color to blue. Reaction on litmus milk, that is stormy clot formation, where lactose, that is milk sugar, can undergo fermentation to acid and gas. This acid coagulates casein milk protein, which in turn forms clot. In presence of gas, it causes stormy clot. Lipase test. The purpose is to see if the microbe can use lipid as a source of carbon and energy for growth. A positive lipase test is noted by the presence of an iridescent sheen, that is oil on water, and clear halo that surrounds the area where lipase producing organisms are grown. Gelatin hydrolysis. Gelatin hydrolysis test is used to detect the ability of an organism to produce gelatinases that liquefy gelatin. Organism producing proteolytic enzyme and gelatinases hydrolyze gelatin into polypeptides and individual amino acids. The bacterial cell can then take up these amino acids and use them in their metabolic processes. In the process, gelatin loses its structure and becomes liquid, even at 4 degrees Celsius temperature. INDOLE test. This test demonstrates the ability of certain bacteria that express tryptophanase enzyme to decompose the amino acid tryptophan to indole, which accumulates in the medium. The bright pink colored ring on the top layer is positive reaction that indicates indole production. The absence of the ring indicates negative reaction. This is a chart which is showing the differentiation of important Clostridia based on their biochemical analysis. Fusobacterium. It is another anaerobic bacterium. The amino acids and peptides are used for energy metabolism. It ferments glucose and maltose with production of acid, particularly the butyric acid. Also produce indole and H2S, but not liquefied gelatin, and is oxidase negative. Thank you for watching. Don't forget to subscribe to the channel and hit the notification bell to remain updated with more such videos.